Okay, in this experiment, we are going to discuss on the. This is also the experiment from the paper 5, that is plant physiology and biochemistry. So, the aim of the experiment is to study the effect of the substrate concentration on the activity of this enzyme amylase. So, as we already know that the enzymes are nothing but they are protein molecules, and these protein molecules, these enzymes, they help in catalyzing the reactions, chemical reactions. So almost all the biological reactions, the chemical reactions occurring in a biological cell, they require enzyme, okay, in order to occur at rates which are sufficient for life. So these enzymes, they many factors, they affect the activity of the enzyme, okay, such as the concentration of the substrate, the pH, temperature, or the inhibitors. Inhibitors are those molecules which will inhibit the activity of the enzyme, and many other factors, they affect the activity of the enzyme. So in this particular experiment, we are going to choose only one factor to see how it affects the activity of the enzyme. And also, you know from biochemistry that the reactions, the reactants, okay, the molecules which react, they are referred to as the substrate. And the molecules which form after the reaction has occurred, they are termed as products. So in this particular experiment, we are going to study the effect of the concentration of substrates, all right, on how it affect the concentration of substrate, how it affect a particular enzyme. And in today's experiment, we will actually study the effect of starch. Okay, we will be studying. We will be taking starch, using starch as a substrate, and we will be using enzyme amylase in this particular experiment. Why? Because just remember one thing, that the enzymes, they are highly specific for their substrates. For example, like in today's experiment, we will be using starch, and this starch is a substrate which can be enzymatically be broken down only by amylase. Okay, it can be broken down into its products such as maltose and other reducing sugars. It can be broken down only in the presence of an enzyme amylase, and not any other enzymes. So just remember this one point, that all the enzymes, they are highly specific for their substrate. So that is why in today's experiment, since we want to study the activity of the enzyme amylase, so we will be using starch as a substrate. So this is the reaction where starch, in the presence of amylase, it is broken down into maltose and other reducing sugars. And also, this I had already mentioned, that it can be affected, the activity of the enzyme can be affected by many factors. So in today's experiment, since we are going to study only the effect of substrate concentration, so what we are going to do, we are going to change only the concentration of the substrate to see that the effect of this substrate concentration on the activity of amylase. But we are going to keep other factors constant. We are not going to change temperature or pH or inhibitors or any other factors. We are not going to change. We will keep them at constant, but we are going to change only the concentration of the substrate. And there are many methods to study the enzyme activity, and this can be done either by measuring the amount of substrate which are consumed during the reaction, or we can study the activity of the enzyme by measuring the amount of the products formed after the reaction. And This experiment, like I had said, that it is about studying the effect of the substrate concentration on enzyme activity. That is why here the activity of the enzyme amylase was studied at different concentration of substrate, that is starch. We are going to use starch, and keeping other factors constant. And this activity of the enzyme amylase, it can be measured by determining the amount of starch consumed in reaction. So now coming to the requirements in this experiment, first we will need the amylase extract, all right, enzyme amylase. Then we have to prepare amylase extract, and then we will be requiring starch solution. Okay, we are going to prepare this starch working solution at this concentration, 1 microgram per ml. And then we will also need I2Ki reagent. This you already studied in the previous experiment, that how starch it will react with I2Ki reagent in solution to give a deep purple color. Right, starch iodine complex, if you remember in the first experiment, and that starch iodine complex we can we can get the concentration of starch by measuring the amount of that starch iodine complex form, all right, by measuring the absorbance. Now we will also need acetic acid. This acetic acid is required for in order to stop the reaction. Okay, after you add amylase enzyme, after the reaction has occurred, then we will stop the reaction by adding acetic acid. Then we will also require other things such as the pipettes, glasswares, mortar and pestle for making the amylase extract, and also we will require a calorimeter for measuring the absorbance of the solution. Now coming to the preparation of reagents. So the first one, that is amylase extract. How you will prepare amylase extract? We are going to take 10 gram of potato. This 10 gram of potato is first crushed into fine paste using a mortar and pestle, and 50 ml of water is then added to the paste. And then after adding 50 ml of water, this solution is then filtered. Then you will take only the filtrate and centrifuge at 3000 rpm for 10 minutes. So after centrifugation, you will see that this pellet, you can throw the pellet and you will take only the supernatant. Then the supernatant obtained is transferred into a measuring cylinder, and the volume is now made up to 100 ml with distilled water. Right, so that will be our enzyme, amylase enzyme. So the next reagent that we require is the starch working solution. Now for preparing the starch, we will require 100 milligram of starch in 100 ml of water. Alright, so that will be the working solution of starch, and you also need to heat the solution until we get a clear starch solution, until a clear solution is obtained. And the third reagent is the I2Ki reagent, and for this we need to prepare 0.6 milligram of potassium iodide at 0.6 gram of potassium iodide and 0.6 gram of iodine. They are dissolved in 10 ml of water, and that will be the stock solution. Then from this stock solution you will take 1 ml and add 100 ml of distilled water. So this will be the working solution. Now coming to the procedure. So in the procedure, what you will do first, you will take, you can see here in test tube racks, you have to take a test tubes and you have to make concentration gradients of starch solutions. Okay, this in the first test tube, as you can see, we are taking blank. We will be preparing the blank, and in other test tubes you are going to make the concentration gradient. So first you will mark all the test tubes with the marker pen. So we are going to start from 0.2, 4, 6, 8 and 1. Okay, and also the blank, and as you can see here I am doing in 2 replicates. So all these will be prepared, the concentration gradients of starch solution will be prepared in test tubes in 2 replicates by diluting the starch solution with distilled water in appropriate proportions, and blank was also prepared with 1 ml of distilled water only. So here what you will add, 0.2 ml of starch solution and 0.8 ml of water. So that will be 1 ml. Here you will add 0.4 ml of starch and 0.6 ml of water. So accordingly you will do in all these test tubes, you will add starch solution and distilled water in appropriate proportions. And after that, the next step is that you will add another 1 ml of distilled water to all the test tubes including the blank to make the final volume to 2 ml. So next we will add 0.5 ml of I2Ki reagent in all the test tubes including the blank. And just remember here the blank, only water is present. Since it is a blank, we will not add any starch solution in the blank. So directly you will add 2 ml of water. Now after adding 0.5 ml of I2Ki reagent, we know that starch will react with the iodine to give a blue colored complex, starch iodine complex. So then after adding after adding I2Ki reagent, we are going to take the absorbance reading. Alright, so that we will know what is the concentration of starch in all these test tubes. You will take the first absorbance reading of all the test tubes at 580 nanometer in a calorimeter. Next, after taking the first absorbance reading, now we will add the enzyme. Okay, so 0.2 ml of the amylase extract is added to all the test tubes including the blank and incubated at 40 degrees centigrade for 10 minutes. Then the reaction will occur. So the starch, which is a substrate, will be broken down into its products. Alright, when you add enzymes, now the starch present in the test tube will be converted into its products. And after incubation time is over, 0.5 ml of 1 normal acetic acid is added to all the test tube in order to immediately stop the enzymatic reaction. Okay, after you add the amylase extract, after 10 minutes you will add acetic acid to stop the reaction. Then a second absorbance reading is taken again at a wavelength of 580 nanometer. Then after getting the second absorbance reading, the activity of the enzyme amylase at different starch concentration is observed by plotting the graph with the starch concentration on the x-axis and the amylase activity, that is, you will use this formula, the difference between the first absorbance reading and the second absorbance reading, OD1 minus OD2 on the y-axis. So this is how you will get the graph. You will see that at first, as you increase the substrate concentration, alright, for example from 0.2 to 0.4 to 0.6, you will see that the rate of reaction also will increases like this. But at a certain point, you will see that the rate of a reaction will become constant. Okay, so what happened, it will become constant at a certain point. This is because, I will just show you this diagram here. You know what happened at first, you can see at low substrate concentration, okay, this red in color, suppose it is a starch molecule, starch concentration. So you can see many enzymes are there, many enzyme molecules are there, so the active sites of the enzymes are free for the substrate to bind and for the reaction to occur. So at low concentration of substrate, since many enzymes are still free, the reaction will occur at a faster rate. But as you go along this graph, what happened when the concentration, when you go along the x-axis, as the concentration increases from 0.2 to 4 to 0.6 to 0.8, what happened? You see till here also, for example, at 0.4, you can see that all the active sites of the enzymes are occupied by these substrate molecules. But still a reaction is taking place at a faster rate. But when the substrate concentration, as you increase the concentration of a substrate more and more, what happens, since all the active sites of the enzymes already are occupied by the substrates, so the enzyme is already saturated. Okay, it is saturated by the substrate than it can deal with. As you can see here, enzyme is saturated far more substrate than it can deal with. So although you add more and more substrate, a point will come that the enzyme will be saturated and the reaction will just takes place at a constant rate. As we know that after this, suppose this is a starch, after it has been converted into its sugar, into maltose and other reducing sugars, after it has been broken down, it will be released from this enzyme. Then this enzyme will bind to another substrate. Okay, it will help in the breakdown of another substrate molecule. So that will go on. So it's not that the reaction will stop, it will continue to go on, but at a constant rate. Okay, it will not be faster anymore, as all the enzymes are busy, are saturated with substrates. Not in this case, at low concentration, you can see that many enzymes are still free, so the reaction will occur at a much faster rate. So this is how you will get a slope, that at low substrate concentration, as you can see, as you increase the concentration of substrate, the enzyme reaction will also increase. But then a point will come where all the enzymes become will become saturated with substrate. So increasing, although you increase the concentration of the substrate, it will, or it does not affect the rate of the reaction anymore. So that is the point of saturation. Now in the observation, you will write everything. Okay, this is the table, how you will write this is the different concentration of starch, then the number of observations, then volume of distilled water added to the test tube, then the volume of the I2KI reagent, and here you will take the first reading. So you will write the absorbance reading you obtain, the optical density reading here in this column. Then here incubation, you have to add the amylase extract. So first this column will come. You have to add the amylase extract 0.2 ml to all the test tube. Then this incubation at 40 degrees centigrade for 10 minutes will come. Alright, first you will add the enzyme and then you will incubate. So this you please write in this next column after this. And the volume of acetic acid is added to stop the reaction after incubation, and you will take the second absorbance reading, and thus the amylase activity can be obtained from the graph by using this formula OD1 minus OD2. So you will take the difference between these two optical density and you will get this column. So this one we are going to plot in the graph. Okay, so this is how you will obtain the graph. As you can see here at 0.2 concentration of substrate or starch, the amylase activity, it increases slowly till 0.6, and you can see from here it starts going down and it starts remaining constant. Okay, so if you take a look at this graph at 0.8 and at 1 ml of starch concentration, the activity of the enzyme, it remains constant. So this is the point of saturation. In our case, it is at this 0.8 microgram per ml of starch that the enzyme has become saturated with starch with substrate concentration. So this is the point of saturation. That is why you can see that even if you add the starch concentration to 1, even if you increase the starch concentration to 1 microgram per ml, the activity of the enzyme, it still remains constant as compared to this low concentration. So here what happened after this point of saturation, that means even if I increase more of the starch concentration, if I add 1.2, 1.4, 1.6, you will find that this slope, it will remain constant. You will get a straight line here. Okay, that means the enzyme activity now will remain constant. So with you can see that with increase in substrate concentration, the enzyme activity also increases until it reaches a point of saturation where the enzymes, they are saturated with substrates, and thus the activity of the enzyme will remain constant. So how you will see in your result, as you can see from the graph that we are getting from the graph plotted, it was observed that at lower starch concentration, the amylase activity was low, and with the increasing starch concentration, amylase activity was also observed to increase, but however, at starch concentration of 0.8 microgram per ml and beyond, there was no further increase in the activity of amylase enzyme, and it remains constant at its maximum. And also you will mention in the discussion that what happened at lower starch concentration, large number of active sites, they are free, and thus they are available for starch to bind, so that when they bind to the enzyme, the starch can be broken down into smaller molecules. But with increasing starch concentration, what happened? More of the active sites, they are being occupied by more starch molecules, which results in increase in the rate of a chemical reaction. But however, as you can see from your result, that at concentration of 0.8 and beyond, all the active sites of amylase, they are already occupied by starch. So no more active sites are available or are free. Therefore, even if you increase the starch concentration more and more, the activity of the enzyme amylase will remain constant. Alright, so this is how you will discuss your result. The conclusion from this experiment is that at this concentration of 0.8 microgram per ml and beyond, the activity of the enzyme amylase, it remains constant and it increase no further.